With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and microcap data place. Welcome to this special edition of the C-Suite series presented by Channel Check and Noble Capital Markets. Noble is an SEC-registered, FINRA-licensed broker-dealer and the source of the equity research available on Channel Check. This investor forum at the World Stem Cell Summit presentation features Caladrius Biosciences, NASDAQ ticker symbol CLBS. Following a brief overview presentation from President and CEO David Masso, Noble Senior Research Analyst Robert LeBoyer will moderate a Q&A session. If you registered for this event through channelcheck.com, in the coming days you'll be given an opportunity to get your own questions answered by the company. With that, I am pleased to present Dr. David Masso.
Hello everyone, my name is David Masso and I'm the President and Chief Executive Officer of Caladrius Biosciences, where we are developing regenerative therapies that reverse chronic disease. At Caladrius, we are looking to bring cellular therapies into the field of cardiovascular medicine, and our company represents what we believe to be several investment opportunities that are attractive to investors. First and foremost, we are a CD34 positive cell therapy platform company, which yields a multi-product development pipeline, and a number of our programs have the equivalent of the Regenerative Medicine Breakthrough designation. Our programs are based on proprietary field-leading technology. We're working only in areas of very high unmet medical need, backed by a strong intellectual property portfolio with patent protection out into the next decade. Important for investors, we have multiple potential value-creating events throughout the pipeline over the course of the next 1 to 2 years, and we're entering 2021, or the remainder of this year, with a very strong balance sheet, about $112 million in cash at the end of the first quarter, with no debt, and a cash runway that's projected to fund current operations well into the future for several years, and we are operated by a seasoned management team. We're working in the area of CD34 cell therapy, and I'd like to provide a technology overview. CD34 cells have a well-characterized mechanism of action. They are naturally occurring endothelial progenitors that are known to reestablish blood flow to underperfused tissues based on a pro-angiogenic and anti-inflammatory mechanism of action. Essentially, they promote the growth of new microvasculature in areas where an ischemic insult has occurred. They've been studied extensively in the clinic, having been being part of clinical trials where more than 1,000 patients have participated, and there's a consistency of the data that is remarkable. We always demonstrate that a single treatment elicits a durable therapeutic effect, and we've never had a cell-related adverse event reported to date. Our manufacturing process for this autologous cell therapy is rapid, it's economical, and it's already scaled to meet phase 3 and early commercial demand. It starts with a pharmacotherapy using GCSF to mobilize the patient, who is the donor, their cells, their CD34 cells, from their bone marrow to the peripheral circulation. There they undergo a apheresis collection process, and that sample of monocytes is then sent to a central processing facility where the CD34 CXCR4 positive cells are isolated, concentrated, and then formulated for appropriate administration back to the same patient at the site, which is directly corresponding to the disease being treated. There's no genetic manipulation or ex vivo expansion of cells. There's no surgical bone marrow aspiration of the cells, and the entire process is about 4 days from vein to vein. We have a strong intellectual property portfolio, as I mentioned, which provides coverage equivalent or comparable to what a small molecule composition of matter claim might give. We have patents granted throughout the world and with fundamental protection at least into the early portion of the 2030s, and with the possibility of expansion beyond that. And that's led to a interesting clinical pipeline, which contains a number of products: CLBS 16 in coronary microvascular dysfunction, HONEDRA or CLBS 12 in critical limb ischemia and Buerger's disease, CLBS 201 for diabetic kidney disease, and OLOGO or CLBS 14 for no option refractory disabling angina. And I'll touch on each of these programs briefly now, starting with CLBS 16. But before I get into the heart programs, I just want to remind everyone that heart disease remains the #1 killer in the developed world, more than all cancers combined, and that recent data published at an AHA meeting demonstrates that there still remains a strong need for treatments beyond the typical and standard large vessel interventions that many patients undergo. CMD is considered a heart microvasculature deficiency without large vessel obstructive disease. It has the same types of symptoms, though, as large vessel disease: frequent debilitating chest pain, but it's not treatable by stents or bypass, and it responds poorly or not at all to available pharmacotherapies. It is a principally female disease with somewhere between 2 and 3 times more females than males experiencing the disease, and it results in the same poor prognosis as large vessel disease, with an increased risk of MACE, major cardiac events, and all-cause mortality. It is a disease that affects a very large number of people, somewhere in the area of 400,000 to over 1.5 million patients in the United States alone. And it is a place where we've done some interesting early work based upon a proof of concept study called ESCAPE CMD, the results of which were reported in 2020, very highly positive, which demonstrated that we, with a single intervention of CLBS 16, could change the physiology of the heart as measured by coronary flow reserve from people being diagnosed as having CMD to being in the normal range, and also demonstrating clinical symptom improvement in a commensurate fashion, taking patients from more than 4 episodes of debilitating angina a day down to 2. We also demonstrated an equivalent statistical significant effect in all of the aspects of the Seattle Angina Questionnaire. And so, we ended up with a very positive trial, which now lets us move to a phase 2B trial, which is ongoing right now. It's a double-blind, placebo-controlled trial that was initiated in the last quarter of last year. It's expected to complete enrollment late this year, although that's possibly going to move into 2022 based upon the impact of COVID on recruitment rates for trials of this type. And we expect to have data in late 2022. And then we'll move on, of course, to pivotal trials should that be as positive as the ESCAPE CMD trial was. Our program in Japan for HONEDRA or CLBS 12 is SAKIGAKE designated. That means it has a special breakthrough fast track regulatory designation in Japan. And we also received an orphan drug designation for Buerger's disease for this product in the United States recently. CLI and Buerger's disease are both arterial obstructive diseases that occur in the lower extremities. They ultimately result in severe rest pain, non-healing ulcers, and typically progress to amputation. And in Japan alone, this represents a multi-$100-million opportunity. For those of you who have never heard of CLI, you can see that when compared to most cancers, it actually has a higher mortality and incidence rate. So it is a serious disease and is one that causes a great discomfort. We're focused on those patients who are considered mild or moderate to severe, but still have functionable, salvageable tissues. And our confidence in the program comes from phase 2 data generated in Japan and the United States, which shows that we can, with a single treatment, convert patients from CLI to being CLI free and have them remain there durably for many years, and in the case of this trial, up to 4 years. And we've also demonstrated a statistically significant improvement in amputation-free survival, which is an endpoint that the FDA typically uses. This has all led to the SAKIGAKE designation in Japan and to the ongoing registration eligible trial in Japan of 37 subjects divided between CLI and Buerger's disease patients. This is a trial that's almost completed enrollment. We've only a handful of patients left to do, but has had a major impact felt from the COVID-19 pandemic. We haven't really been able to enroll a patient in the trial for about 18 months as a result of the states of emergency that have been implemented in Japan and then extended and continue to be extended. So once those states of emergency are open or lifted, we should have enrollment complete, we think, within a quarter. And so far, data in this open label trial is trending positively, and we can talk about the Buerger's disease cohort, which has been completed for some time, where we've shown a 60% cure rate, to use that word loosely, changing those patients to CLI-free in a disease where the natural patient evolution is for every patient to go on ultimately to amputation. So far, very promising, and we'll look forward to that. We're also discussing the program with FDA, and we'll see if there's a path to registration in the United States for CLBS 12. Moving on to 201 and kidney disease, you can see that kidney disease is a growing problem here in the United States, and one that requires a fair amount of focus. We'll be looking at patients who are in the progressive stage, but prior to kidney failure, so stage 3/4 patients, probably focusing on the latter stage 3B and early stage 4 patients, to demonstrate not only the ability to stop progression of the disease, but hopefully to regenerate the kidney and to improve GFR, which is something that we haven't seen a regenerative medicine do yet. And so we'll be looking to do that in this proof of concept trial, which is currently being designed. We have filed an IND and have it open with FDA. We're finalizing the protocol and hope to be enrolling patients by this fall in a trial that preliminarily looks like the synopsis that you have on your screen, but the exact protocol remains to be finalized. And we'll look forward to enrolling that program and having data probably in late 22. And then finally, I'll just mention OLOGO, our program for no option refractory disabling angina, which does have an RMAT designation in the United States. Again, breakthrough/fast track benefits. It's a program with a lot of clinical data, but where we've run into a bit of a roadblock with FDA in that the agency has agreed that a single trial based upon the data that's been generated to date for phase 3 would be acceptable, but they're requesting quite a large trial in an area where there are less than 100,000 patients, probably less than 50,000 patients available. So it's an orphan-sized disease where a 400-patient trial, especially with 50% of those patients going into placebo or standard of care, could take a very, very long time to enroll and probably not be practical. So we continue to work with the agency to see if there's a way that we can reduce the size and scope of a phase 3 program that would lead us to registration, but still be practically something that could be implemented and affordable. So, a recap quickly, the milestones for the programs are shown here. I'll just remind everyone that the pandemic continues to take its toll on our ability to predict accurately enrollment rates and completion. And so all of these are based upon our best guess today, but could change dramatically as COVID-19 progresses, especially with the new Delta variant coming into play. I'll just mention quickly, our financial situation is quite stable, very simple cap structure, and about $112 million in available cash at the end of the first quarter, with a roughly quarterly burn of about $8 million. But that's based upon the normal projected clinical enrollment. It could actually be a little bit less than that if clinical enrollment lags because of COVID-19. And then we come back to the summary of the company. But overall, a company that has strong fundamentals, has actually done quite well during the pandemic in terms of raising additional capital, keeping our programs on track, and actually initiating new programs. But like all companies, we're suffering from the inability to project enrollment rates and target completion dates simply based on the uncertainty that the COVID-19 pandemic brings to clinical trial execution, especially for trials like ours, which require hospital space. So I'll stop there and look forward to moving now into the Q&A section with Robert LeBoyer. And thank you for your attention.
Thank you, Dr. Mazo. That was a very interesting presentation, and it was nice to hear about all the progress that you've been making. One of the things that you mentioned were the challenges that COVID-19 has brought both domestically and in your Japanese trials. Could you discuss a little bit more of the challenges in executing those clinical trials and what you're doing in response?
Certainly. And again, Robert, thanks for inviting me to participate to the conference. We're really pleased to be here. So COVID-19 has provided a number of obvious challenges, right? I mean, if patients are infected or actually sick, they can't participate in a trial. So that much is pretty obvious. But what is an added complication for a trial such, or the trials that we conduct, is that our trials require at some point, if not admission to hospitals, certainly the use of hospital space, right? So when you're looking at a program which requires a patient to receive GCSF injections, that can be done in an office or even at home, but then to go into a place to go through an apheresis, which is like a dialysis. It's not really a terribly complicated procedure, but it's often done in a clinic or a hospital. And so, those spaces are typically not available because of COVID-19 restrictions, or they are, doing much more testing and adding many more layers of call it health security to allow people in and out of those facilities, and it just makes it more complicated. On top of that, our Japanese trial requires that once a patient is treated, that they need to be observed for 5 days in a hospital setting. That's got really nothing to do with anything but a regulatory requirement. It's got nothing to do with safety or anything else. But because of the states of emergency in Japan, all the hospitals are restricted to only COVID patients. So while we could treat a patient, we don't have a bed for them to be observed in, and so then we can't enroll patients. So this has been, part of the challenge when doing trials that involve interventional type procedures, and I think we'll be getting over that sometime soon, but it has caused delays and it makes it really difficult for projecting enrollment rates.
Yes, we've seen that throughout the industry, and like you mentioned, the health safety and all the precautions have slowed down the number of office visits. And in addition to non-essential treatments being stopped, even the essential ones that require a hospital visit have slowed and been delayed because of all the extra precautions, staff cutbacks, and just a general across-the-board reduction in medical services.
Exactly.
So what you're saying is not a surprise.
Mm-hmm.
So many of the cell therapies that are in development have shown promise early on, similar to your programs, but then faltered in later stage clinical trials. What gives you the confidence that your CD34 cell technology is both safe and can be efficacious and successful?
That's a really good point, and I appreciate you bringing that up, because we suffer, as do a number of our I'll call them colleague companies who take a similar approach. We suffer from what I would characterize quite simply as a fair amount of bad science that was done in the regenerative medicine field early on by a variety of academics and even other companies, where they jump to conclusions too early, where they would do a very small study in a very select group of patients at a single place and then extrapolate that to broad worldwide applicability in a very important disease state, and never be able to translate that further into development. It was a very naive approach. We and a number of other companies like this have taken a much more rigorous approach, akin to what you would do for the development of any kind of approach, treatment or molecule. But that approach has, it takes more time, it takes more money, and it takes more studies, and people get frustrated, because of that. What gives us the confidence to continue, and I think what has given us the ability to raise, the significant amount of money to back our programs, is that we are doing things in a very methodical fashion. We're answering key questions of development, and we're focused on what it will take to bring these products to patients in a commercial setting. So these are not done for academic reasons. These are done to help us provide new treatments for patients in need. And so far, as I said, we've had large numbers of patients exposed in clinical trials using CD34 cells, and there's never been a cell-related adverse event reported, which is what you would expect for an autologous cell therapy. People get their own cells. So safe, I think everyone takes as a given now. Efficacious is one that also, I think, has a strong level of confidence because of the amount of human clinical data that we've generated. So we've had hundreds of patients tested across a variety of indications, and we always get very strongly positive results of therapeutic effect or statistically significant, effects that point toward efficacy. So I think that's what you would look for in the development of any therapy. It's especially important in a new type of therapy like cellular therapy, and it's something that a lot of other companies didn't have previously that we do have. So that's why we have the level of confidence that we do.
Well, yes, I agree. And having followed the field of cellular therapies and regenerative medicine for many years, I think in the early days, there was a lot of euphoria and a lot of high expectations that this was going to be a great cure for many things. And those expectations were very high. Like many new therapies, the initial stages of research narrow down those indications to things that are more practical and more justified. But there's a big disappointment that this wasn't the cure for everything. And
right.
then a lot of companies will stop their programs, but the ones that actually have data go forward and come up with real plans to reach the market. And I think that's the stage that you're in, where you have data and you are at the stage where you can actually have a plan to reach the market with FDA interactions, and this isn't going to cure all of man's illnesses.
Right.
You're going to be very specific to specific illnesses that will do well for the company and those diseases in patients.
Exactly, and I think the two regenerative medicine regulatory special designations that we received, the SAKIGAKE designation for CLBS 12 in Japan and the RMAT, or Regenerative Medicine Advanced Therapy designation for CLBS 14 here in the United States, I think underscore the rigor of the approach, because you don't get those without human clinical data. And not everybody gets them. So, it's an important, I think, check box to look at to see that the regulators agree that we're on a path that is logical and toward treating things that currently have no treatments.
Yes, and early on, when there were many companies rushing into the field, nobody had those things.
Right.
And now you have data to show that these things are scientifically justified, and the regulators agree. So that narrowing of the field and being more specific to the things that you can be successful at is actually a positive.
I agree.
Okay. So speaking of the field and the technology, could you discuss the scientific rationale for expanding the pipeline and additional applications?
Sure. So we're actually taking, like most biopharmaceutical companies, you have a principal or core technology that you look to develop, and then you always are looking for, I would say, complementary technologies to bring in and to add, because all of us have been doing this. I've been doing this for almost 35 years, right? Drug development. We all know that despite the rigor, the best intentions that we have, the strong data that you get, at various stages of the program, that there's a very high attrition rate in drug development, and so it's always good to have multiple things being developed. So within our company, we look first for diseases where there is a high unmet medical need, where a treatment which fundamentally provides new microvasculature, reperfuses damaged tissues, could have a major effect. And so obviously that, what jumps to mind are cardiovascular indications like angina, maybe stroke, which we're not doing anything in at the moment, but would be another area where we could venture. And then you start looking at other organs which are damaged by ischemic events or the loss of blood flow. And of course, diabetic kidney disease or kidney disease in general, jumps to mind. Diabetes with critical limb ischemia, peripheral arterial diseases, and so on and so forth. So we continue to look at that, and that's how CLBS 201 was born, because we've looked now at attacking deficiencies in the microvasculature of the kidney as the next frontier after we've been generating positive data in the heart. But there could be applications in the lung, in the liver, and in other organs. And then, for expanding the pipeline, of course, we look more broadly for complementary technologies, regenerative or otherwise, that could be in a similar therapeutic area or in a similar disease specifically. And and if we can acquire those or collaborate with others in those areas, I think we'll try to do that as well, all with the idea of maintaining a rich pipeline of possibilities for the benefit ultimately of patients in the long run, but also for our shareholders.
Speaking of all these different applications, one of the things that you mentioned is that your cell collection process is much less invasive than others that I've followed and that I'm aware of, but your mode of administration is somewhat more complex. Could you discuss that a little bit and just bring these two things into a little more detail and what would be required clinically?
Yeah, so let's start with the collection side. We have a very much optimized collection process here with the pharmacotherapy using GCSF to mobilize the cells so that no surgical aspiration of bone marrow is necessary, an apheresis, which is relatively non-intrusive, and then a quick processing of the cells that are then returned to the patient for injection. We've also been working on, we've got data that shows we can cryopreserve the cells so that if we don't use all the cells collected from a single donation, we could freeze them and use them at later dates for the same patient, of course, if that ever became necessary. So on the collection side and manufacturing side, we've optimized things and have plans for further optimization. On the administration side, we started initially with the idea that to help Mother Nature along, it would be best to inject the cells close to the area of ischemic insult. So if you've got a patient who has CMD, for example, that's coronary microvascular dysfunction, meaning their heart muscle doesn't have enough capillaries, then you'd probably want to inject them into the vasculature of the heart, if not the heart itself. And so we started with intracoronary injections in CMD. And so far the data there shows very promising results. So we'll continue with that. With NORDA, which was one of the earlier applications, we started with an intramyocardial injection. That's an injection right into the heart muscle, much more complicated, right? And ultimately, we're looking to move away from that because we think that that's the most intrusive and the one where the procedure provides the highest level of risk. CLI is a simple intramuscular injection, right? And the kidney program will start with an intraarterial injection into the renal artery, but ultimately we'll try to do some work with retrograde or intravenous injections to see if simply putting the cells into the circulatory system in a concentrated fashion is enough to let them find their way to the area of insult, because no matter where you inject them, they enter the circulation anyway. But right now, the data seems to indicate that putting them proximal to the ischemic insult makes sense, and so we'll continue to do that until we can progress to something simpler.
And just turning to some of the financials, could you repeat how much cash you have and whether you anticipate raising capital anytime soon? Or
Well,
if you did, what you would do with it?
Yeah, we raised about $90 million in the first and early second quarter of this year, all during COVID, right? And all working remotely. So I think that shows that there was a great deal of appetite for people getting into this type of technology. I'm glad we did, because since then the market for small cap biotechs has become a bit more challenging. And so I'm very pleased that we were able to take advantage of that and put a lot of cash in our balance sheet, which secures the financial future of the company for the next at least 3 years and maybe beyond. So we're in very good position that way. As a result of that, we certainly don't have any plans to go out and raise money at the moment. But I would be remiss if I said that if the offer for capital became available and the price was right, certainly we would seriously consider it and probably take it, because drug development is expensive. There are more things that we could be doing. With more money, we could probably do them faster. And so you never really have enough capital. And our goal is, of course, to get products ultimately to market so that we could be self-sustaining in that regard. But until we do, we'll have to raise money. No plans for the foreseeable future to do so, but I wouldn't say no to capital offered under the right circumstances.
Well, yes, it sounds like you have more good ideas to do research than money to fund them. So that's a good problem to have.
Exactly.
Any closing remarks or anything you'd like to add? This has been a really interesting discussion, and it's really nice to see the company moving from that early stage where there's a big flow of interest in the speculative technology, the one that's really backed by science and has a clear route to the market.
Well, I think that, and the only thing I'll say is, again, thank you for letting us participate. And people can contact us through our website or through our head of communications, John Mendito, with any specific questions. But if you look back to the presentation, the summary slides really, I think, tell the story in a nutshell. We are a company with strong fundamentals. We're executing well. We've been transparent since this management team took over, which is now going on 5 years. And I think we've developed a level of credibility for doing what we say we're going to do when we're going to do it. So I hope that people will remain interested and follow us and hopefully become shareholders and share in our success. So thank you again for inviting us to participate.
Thank you for joining us for this C-Suite interview presentation brought to you by Channel Check. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and micro-cap companies listed.